Two-letter acronyms can only be understood if there is context to help. PD can mean many things. In the context of drug development, it may mean protocol deviation. In the context of oncology drug development, it may mean progressive disease. Don't mix that up with PD-1 or PD-L1. Those are drug targets in immuno-oncology. In other medical contexts, PD may mean Parkinson's disease, that is a neurological disorder with shaking hands, peritoneal dialysis, which is done for short periods when the kidney function needs to be substituted, or personality disorder. I was a PD for a couple of years. In Germany, that is an academic title. And if the D is a small case d, then PD may mean palladium. Screenshot this as my collection for PDs if that helps. We will move on to drug development. Welcome everybody! You have dialed in a series of teaching videos for drug development in oncology. We are currently doing a series of acronym explanations, and today we have picked PD. My name is Johannes Wolff. This is recorded in January 2025. If you stay on for 5 minutes, you will hopefully learn something, even when you are a seasoned drug developer. In the context of oncology trials, PD means progressive disease, and it is not a good thing. It is an endpoint, a response category used in early oncology trials, and what that means is we have a patient that has a tumor, we give a novel cancer treatment, the tumor responds, and if the tumor completely disappears, we call that time point response a CR, complete response. If it's smaller, we call it PR, partial response. If it's unchanged, we call it stable disease, SD. Or if it's larger, the most unfortunate case, then it is called PD. These categories are described in more details specific for different cancers in various response criteria publications. It is a very interesting field, and we have many videos about it. One point is sort of common among all of them. The term PD-dogma is something that belongs in the past. When you treated a cancer and the cancer get bigger, then the typical teaching of oncology is you should stop and you should try something else because the treatment doesn't work. That means, in the context of a clinical trial, if PD has occurred, there will be no further time point responses. And that was how it was in the past. That is what the PD dogma is. But it's not a good thing. Because in modern medicine, we have learned that these assessments are sometimes wrong. The imaging may show the tumor by whatever reason bigger than it really is, or more relevant, the tumor actually may be bigger, but shrink later. And the original looking bigger comes from inflammatory cells or edema, things that infiltrate the tumor without the tumor cells themselves being more. The worst thing you can do in that case, when the drug is working, is to stop the drug. You should carry on, continue measuring responses, and the further time point responses are then, if you're lucky, partial response or even turn into complete response. For that, the protocol needs to allow treatment beyond progression. Now that is what modern protocols typically allow. And if you have a different protocol, then it's a good idea to propose to allow treatment beyond progression in the next protocol amendment. We describe our clinical trials in protocols. That is the document that tells the doctor and the nurse and the patient what to do, at what time point, what drug to give, what procedures to do. If the protocol is conducted and something does not go completely following the protocol, we call that a PD. Protocol deviation. Now you may think it is best to run a protocol without protocol deviations, and of course that is true. But protocol deviations are reality, and protocol deviations are not a crime. It is important to report them and to learn from them. The regulation around that has changed a bit. In the beginning, there actually wasn't much of regulation. There were SOPs, institutional guidelines. When I started at MD Anderson Cancer Center about 20 years ago, they taught me there are violations and there are deviations. And then I asked, which one is worse? And they said, oh, everybody asks that question. Violations were worse. And later this nomenclature was dropped and it was substituted with deviations and classified in major and minor. Violations, or the later term of major deviations, were those that affected either the safety of the patient or the integrity of the data. Just those 2 points, nothing else. And those are fairly high-level umbrella points. They're not quite clear always. If they apply, you needed to think about it. A little later, further classifications came around, still used in some places as critical, major, and minor. And critical are typically those that affect safety. Now, there is new guidance. The FDA has published a draft guidance only in December 2004, so only a month ago for this, when this video is made. They go a bit away from these high-level concepts and go more concrete. They have a chapter for each of those topics. I have lined the chapters up in the timeline. It happens in a protocol. So eligibility criteria, inclusion-exclusion criteria describe which patient can go on a protocol. If those are violated and patient goes on protocol that should not be on, obviously that is an important deviation because any data you find then is not for that disease. It was really meant to be and doesn't mean much, makes everything very complicated. Consent problems are important. Remember that, important deviations. As you know, patients need to be informed before they can enroll in a clinical trial, and then they need to sign. That signature is called the consent. If something is wrong with that, that is an important deviation. Randomization refers to what is typically called the phase 3 studies, at least mostly, where patients are allocated to treatment groups. This may be 2 different drugs or 1 drug or a placebo, which is basically no drug, just looks like a drug. And that randomization is secret. The patient doesn't know, the doctor doesn't know. There are many good reasons behind it. If something is wrong with the randomization, that can screw up all the data and it is an important deviation. Obviously, when the patient gets the wrong treatment or the wrong dose, that is very important. Now, there is no very important. It's an important deviation. If there is an effective cancer drug and you give too much of it, you can hurt the patient with the drug. If you give too little of it, the cancer will grow and that will hurt the patient. Neither of the two is good. And any data that comes out of a wrong treatment does not reflect the treatment that was to be studied, an important deviation. If procedures are missed or not, or the wrong procedures are done, monitoring is missed, that can affect safety. Prohibited concomitant treatments are on the list of important deviations. Those are typically drugs that interfere or can potentially interfere with the study drug, make the level of the drug go up or down. And again, that is almost the same as giving more or less of the drug. And if something is wrong there and prohibited medications were given, that is considered an important deviation. Withdrawal criteria missed are listed by the FDA as important deviations. Withdrawal are criteria that are described in the protocol. Typically, the cancer grows and it is obvious. It's not pseudo-progression, it is really progression. The patient needs to come off, needs to get something else. Or there are severe side effects. The patient needs to come off to protect them from the toxicity of the drug. Or it maybe not even anything with that patient, but the information from other patients comes in and indicates that something is dangerous or not working, and the study has to stop, and you forget that and you don't do that, and the patient continues to get the drug. That is missing withdrawal criteria, and those are important deviations in the eyes of the FDA. The FDA also has some important deviations that are around the data. So if the data around the major study endpoints, particularly the primary endpoints, are somehow screwed up, that is an important deviation. the protected health information is violated. That is an important deviation. Protected health information are things like name, date of birth. Anything that can identify the patient is typically protected and has to stay secret. Not even the sponsor typically knows who the patient is. And if that information leaks, that is a violation of the rights of the patient and an important deviation. And finally, unblinding is listed by the FDA. Now, blinding is something that has typically something to do with randomization. When a treatment protocol is written such that some patients get treatment, other patients get placebo, which is no treatment, so that we can later find out if the treatment actually works by comparing two patient groups. That type of study design is the most powerful way that we have to identify the effect of a drug. And if either the patient or the doctor or both of them actually know which one is the placebo, then the results need to be taken much more cautiously. And if that information leaks in studies where it shouldn't be there, that's called unblinding, and it is an important deviation. Now, that's all described by the FDA in December 2004, only in a draft document. But the FDA isn't all there is. The FDA that stands for Food and Drug Administration, another abbreviation to learn. But it's only for the US, United States. And the equivalent in Europe is the EMA, the European Medicines Agency. Look under the term serious breach. And you will find similar definitions. A serious breach is something that affects safety, the rights of trial participants, those are the patients, and or data reliability or robustness, the old term of integrity of the data. Now, let's learn a few more abbreviations. This is all very important for our CRAs, our clinical research associates, and the study managers at the site. They document PDs, protocol deviations, in the PD log, a document that exists typically with the sponsor and with the site. Sometimes it is in the binder of the patient. It's defined by the institutions. And the group in the institution, the oversight from the institution, from the hospital that looks at that, is called the IRB, Institutional Review Board. That's the American version of it. In Germany, the equivalent is called EC, Ethics Committee. Sometimes, when deviations happen multiple times and you need to do something about it, for instance, add a description, add teaching for the sites, or exclude certain types of patients, then we start a CAPA, a corrective and preventive action plan. And sometimes if it's really bad and we need to do something really urgent, such as stop the trial, drop the dose, add a diagnostic procedure to protect the patients, we call that an USM, an urgent safety measure. And finally, when the entire protocol is done and the report is written, then these major, now important deviations are part of the CSR, the Clinical Study Report. And those are now enough abbreviations. And these were enough acronyms for one video. Our time is up. This video was brought to you by Oncology Consulting Wolff LLC, which is solely responsible for the content of the video. Also, all mistakes that you find in there are my mistakes. Nobody else is responsible. There are many more of these videos. Please check out the channel and please forward the videos to people that might benefit from them. They are free to be watched and welcome to be shared. There are no financial sponsors and the content of the video was completely independent. And we hope you enjoyed it. Bye bye.